test for the detection of HCC in patients with cirrhosis.
Thank you. I'd like to thank the session chairs and also the organizers on behalf of my colleagues at Helio Genomics to be able to present the CLIMB trial, which is a first of its kind prospective blinded multi-center trial in the United States to evaluate a blood-based test for the early diagnosis of hepatocellular carcinoma in patients with cirrhosis. So I don't think I need to explain to this audience the unmet clinical need in HCC surveillance. In the United States, hepatocellular carcinoma is one of the only cancers whose incidence and mortality are steadily increasing. In part, this is due to a large population of patients with cirrhosis, approximately 800,000 in the United States, that have an annual incidence of developing hepatocellular carcinoma that's up to 8%. So based on the recommendations of expert bodies like the American Society for the Study of Liver Disease, these patients are recommended to undergo surveillance at 6 months intervals using ultrasound with or without determination of alpha-fetoprotein. The problems with this strategy are several-fold. One is that the penetrance or the adherence of patients to the screening regimen is abysmally low, at least in the United States. In some real-world studies, less than 10% of patients get guideline-adherent screening. Ultrasound itself has several problems. It is costly. It is not available at many rural care sites in the United States, which may contribute to the low adherence rate. It is also performing not as well in patients with obesity, which is a complication that's shared by many patients with cirrhosis. And lastly, it has low sensitivity for small liver lesions. So additional methods for HCC detection are needed to overcome these deficiencies. The approach of the Helio liver diagnostic test is a simple blood-based test where peripheral blood phlebotomy is done, and then the sample is analyzed in two parallel pathways. In the first pathway, the methylation status of a set of genomic DNA markers is assessed in cell-free circulating DNA using next-generation sequencing technology. In the second part of the assay, the blood levels of several protein biomarkers, including AFP, the AFP-L3 percentage, and des-gamma-carboxyl prothrombin are determined by immunoassay. These quantitative data are then fed into an algorithm that was developed by machine learning using a large data set of cirrhotic patients. And the actual test result is qualitative. It's reported out as either positive or negative. So this is the design of the CLIMB trial, which again is a prospective, multi-center, blinded trial. Patients with cirrhosis are enrolled, and all patients at enrollment get 3 different tests. They get the Helio Liver DX test, ultrasound, and also determination of AFP. All patients then go on to have multiphasic MRI with contrast, and this is used to determine the clinical truth of the HCC diagnosis. The MRI results are binned by liver imaging results in data systems, the LIRADS systems, according to their number. So LIRADS 1 or 2 are scored as negative, LIRADS 4 or 5 as positive. Patients with LIRADS 3 MRIs are determinate, and all indeterminate patients are subjected to a follow-up MRI at least 6 months later to clarify their disease status. This is just the characteristics of the subjects in the trial. The median age was about 60. As in many of these trials that we've heard about, there's a male predominance. About a third of the patients had Hispanic ethnicity, and about half the patients had a comorbid condition, including either obesity or diabetes. The study was conducted both in academic settings and community-based centers, and there was a variety of underlying conditions that contributed to the diagnosis of cirrhosis. So this is just the disposition of the subjects in the trial. Initially, 1,968 participants were consented for the trial. The first 412 patients who were enrolled prior to June 20th, 2020 were assigned to a training cohort to refine the test. And then the remaining 1,556 subjects were assigned to the trial itself. Of these, 271 were not evaluable, and this was principally because they did not get either the ultrasound or the MRI at the initial visit. That left 1,285 patients for the trial itself. And of this group, there were 46 new hepatocellular carcinoma diagnoses made, which represents a prevalence of about 3.6%. These are the characteristics of the tumors that were identified in the trial. Most patients had a single lesion, and the characteristics suggested that most of these lesions were early disease, and that's based on the fact that 74% had small lesions of less than 3 centimeters, and about 59% had AJCC stage T1 lesions. So the Helio Liver DX test met its primary and secondary endpoints and also an exploratory endpoint. This graph on the left shows the sensitivity versus increase in sensitivity versus ultrasound. So the primary endpoint was to achieve at least a 5% increase in sensitivity versus ultrasound. The data was analyzed according to bootstrap analysis to generate a point estimate for the difference, and that was 20% increase with the 95% confidence intervals illustrated here, whereas tests for significance were based on either chi-squared or Fisher's exact test as appropriate. The test was also felt to be equivalent in terms of specificity to ultrasound, and that's based on a 10% non-inferiority margin. In the secondary analysis for lesions that were less than 4 centimeters, Helio Liver DX test also showed an increase in sensitivity of 24%. And then lastly, in an exploratory analysis for very small lesions, less than 2 centimeters in size, this increase in sensitivity was approximately 29%. These graphs just show the test characteristics for the Helio DX liver test on the left versus ultrasound alone, AFP alone, or the combination of ultrasound and AFP, where a cutoff of 20 nanograms per mil was used for the AFP. And again, you can see the superiority and sensitivity of the Helio Liver DX test versus ultrasound alone or the other modalities. This just summarizes all the test performance characteristics, and I would like to point out that the Helio Liver DX test maintains its superiority in sensitivity and specificity over ultrasound and AFP when the cutoff was reduced to a more stringent level of 10 nanograms per mil. Now, if you look at the performance of these tests broken down either by the lesion T-score on the left or the size of the largest lesion on the right, you can see that for lesions of T1, according to AJCC testing criteria, the Helio Liver test had a higher sensitivity, almost 45% versus ultrasound. And then if you look at lesion size, for lesions that are less than 2 centimeters, the Helio Liver test had a sensitivity of about 26%. And in this study, ultrasound failed to detect any of these small lesions. The sensitivity performance was maintained for liver lesions that were between 2 and 3 centimeters, but for larger lesions, the performance of ultrasound and the Helio Liver test were roughly equivalent. The next several slides show analysis by subgroups, either in this slide, the etiology of liver cirrhosis, and in subsequent slides, the race or the ethnicity. And the numerical values for sensitivity for the Helio Liver test exceeded that for ultrasound in all these subgroups, but the study was not powered to detect significant differences. This is just a subgroup analysis by race. And here it is for ethnicity, for Hispanic background, and the white subjects in the test. So just to summarize, the CLIMB trial met the co-primary endpoints for superior sensitivity at a 5% margin and non-inferiority for specificity at a minus 10% margin compared to abdominal ultrasound. And the Helio Liver DX test was more sensitive than ultrasound, particularly for the detection of very small lesions, less than 2 centimeters. So these results suggest that the Helio Liver DX test is poised to become a new standard of care for the surveillance of hepatocellular carcinoma in the United States. And due to its expected increased adherence to screening and its superior ability to detect early small hepatocellular carcinoma lesions, it is expected to lead to improved outcomes, including overall survival, and potentially to reduced healthcare costs. Lastly, the technology that Helio Genomics has used here is now being applied to explore whether this test can be used to monitor the response to disease, including minimal residual disease, and also to predict what type of treatments patients might best respond to, particularly to immunotherapy and checkpoint inhibitors. I'd like to close by thanking all the investigators in the CLIMB trial and the patients who participated in the trial, and also for the audience for your attention, and I'll be happy to take questions.
I'm Stillman, ECU, North Carolina, USA. Do you have data on communal versus academic centers? Because ultrasound is very dependent on who does it. And any screening that is getting done is better than a screening not done. So I congratulate you on the essay, but you know, in your institution, an ultrasonographyst may still be better than the test.
That is an excellent question. As you can see from one of the tables, actually the majority of the patients were accrued at community centers. All the ultrasounds were actually reviewed. They were obviously reported out at their own centers, but they're all reviewed centrally by a blinded radiologist to try to eliminate differences in the characteristics of the ultrasound at the different sites.
Congratulations on a very comprehensive study. When I hear AI, I get a little bit nervous what black box it is. Can you tell us what kind of variables go in? And what mechanism, what specific algorithm was used, and whether how to adjust for overfitting that might be there that makes you overestimate the sensitivity of the test?
So the algorithm was based upon analysis of hundreds of potential methylation sites, and the optimization was done choosing those markers that had the most predictive value for the diagnosis of hepatocellular carcinoma versus other non-malignant causes of liver disease. I can't tell you right off the top of my head about the details of the stringency of the test, but it had been defined initially based upon a group of samples from China. And one of the things that we realized was that the etiology of liver disease in the United States was different enough that it required basically a different set of markers and a different optimization protocol. So that's what we did.
Thank you very much. We can